Assalamualaikum and welcome to my final year project presentation. My name is Balkish Nadira binti Zahlan, a final year student of Bachelor of Science, Honour Applied Microbiology in UiTM Shah Alam. The title of my FYP is Antimicrobial Activity of Lactic Acid Bacteria Isolated from Fermented Durian Flesh, which is Tempoyak, against pathogenic bacteria, and I was supervised by Puan Irne Aileen Rizlan Rus. This is the overview for the presentation, which consists of 8 parts. Now we will move to the introduction. Firstly, we need to know the characteristic and importance of LAB. LAB are gram-positive, non-motile, acid-tolerant microorganisms, including genera like Lactobacillus, Leuconostoc, Pediococcus, Lactococcus, and Enterococcus. Lactobacillus, the largest LAB genus, thrive in carbohydrate-rich environments in human and animal gastrointestinal tract, also producing lactic acid used in food preservation and biodegradable polymers. Next, we will move to the antimicrobial properties of lactic acid bacteria. LAB produce antimicrobial agents like organic acid and hydrogen peroxide, lowering pH to inhibit pathogens. Their metabolites such as lactic acid, acetic acid, ethanol, and bacteriocins are effective against various microorganisms. Next, LAB can act as probiotics. LAB are common probiotics that improve host health by producing antimicrobial compounds like bacteriocins and organic acid, which protect against pathogens. Lastly, bacteriocin of LAB. Bacteriocin. Antimicrobial peptides from LAB are effective alternatives for antibiotics combating antibiotic resistance and pathogens. Now, we will move to the problem statement of this research. As we know, antimicrobial resistance in bacterial pathogens presents a significant challenge, leading to high rates of illness and death. The emergence of multi-drug resistance in both gram-positive and gram-negative bacteria complicates the treatment options, making conventional antibiotics often ineffective and in some cases entirely unusable. Lactic acid bacteria produce various antimicrobial compounds like organic acid, hydrogen peroxide, and bacteriocins, which could serve as alternative to antibiotics. This study aims to investigate the antimicrobial efficacy of LAB, especially against pathogenic bacteria such as Staphylococcus aureus and Escherichia coli. Now we will move to the research questions. The first question is, what are the total lactic acid bacteria count from a 1-week-old tempoyak? The second question is, what are the biochemical characteristics of the isolated lactic acid bacteria? And third, will the selected lactic acid bacteria isolated exhibit antimicrobial activities? Now we will move to the objective of this research. The first objective is to determine the total number of lactic acid bacteria from a 1-week-old tempoyak. The second objective is to study the biochemical characteristic of selected lactic acid bacteria isolated from a 1-week-old tempoyak. And the last objective is to evaluate the antimicrobial activities of selected lactic acid bacteria against S. aureus and E. coli. Now I will move to the research methodology. The first method is the isolation of lactic acid bacteria. Firstly, the tempoyak sample were collected from the local market in Kluang, Johor. Next, 1-week-old tempoyak blended with 0.85% of NaCl solution. Then the blended sample added to the MRS broth in a flask. Then the sample were incubated at 37 degrees Celsius for 48 hours. Then after the incubation, serially diluted the culture up to 10 to the power of 6 times. Then each dilution spreads on MRS agar plates. Then incubate the plates at 37 degrees Celsius for 3 days. Then, the LAB colonies will be counted to determine the total lactic acid bacteria count from a 1-week-old tempoyak. Then lastly, individual colonies pick and stick onto the fresh MRS agar plates to obtain single colonies. Next, for the second method is biochemical test of lactic acid bacteria. The first test is gram staining. Second is carbohydrate metabolism test. Third is catalase test. Fourth is SIM test. 5 is MRVP test. Next, for the last method is antimicrobial assay, which is agar dish diffusion. Firstly, the agar plates inoculated with S. aureus and E. coli by streaking the entire surface. Then, place sterile filter paper dish on the agar, and LAB culture added to each dish. Then, the plates were incubated at 37 degrees for 48 hours. Then, the diameter of the clear zones measure around the dish, which indicates the antimicrobial activity of LAB. Now, we will move to the significance of study. Firstly, the development of new antimicrobial agents. Investigating LAB from tempoyak can lead to discovering new antimicrobial agents as alternative to antibiotics. Second, combating antibiotic resistance. LAB could provide solution to the serious threat posed by widespread antibiotic resistance. Lastly, alternative to current antibiotics. LAB may serve as effective alternative to modern antibiotics for treating bacterial infection. Now, we will move to the last part of this presentation, which is the expected outcome of this research. The first expected outcome is identification and discovery of antimicrobial characteristic of LAB isolated from tempoyak. Next is the biochemical characterization of LAB will identify their characteristics and differentiate them from other bacterial groups. And lastly, the study will demonstrate significant antimicrobial activities of LAB against pathogens such as S. aureus and E. coli.